Assalamualaikum everyone, welcome back to this channel. This is me Arya Wahid and you are on the channel of Medicare. So as you know that we have started the lectures about the anti-cancerous drugs. So we have discovered some other classification. Now we are going to start the alkylating agent that are very similar to the platinum coordinate complexes. So these two classification are similar to each other. Alkylating agents, what are them? Let's study them. They are non cell cycle specific. So let's study them, that what they said in this. So now, yeah, alkylating agents, they exert their cytotoxic effects by covalently binding to the nucleophilic groups present in your cell. So actually they bind with your DNA and the RNA and produce cytotoxic effects that are very lethal to the tumor. And they can be act on your normal cell or the toxic cell, mostly toxic for the rapidly dividing cells. Now first classification, and these are covalently bound to your mainly DNA and leading to alkylation. So they add alkyl group into your DNA, causing its damage. They are mutagenic and they are carcinogenic. Types of alkylating agents. So the first type is the nitrogen mustard drugs, which are cyclophosphamide or isophosphamide. These two drugs are considered as mustard gas or also called as nitrogen mustard. So their mechanism is, so they activate, first of all they will activate with the CYP450 enzyme that is present into the liver, and they convert into the active metabolite. First metabolite of them is the phosphoramide mustard, and second metabolite is acrolein. First metabolite, which is the phosphoramide mustard, this is cytotoxic. This is the major metabolite that will perform its action. If we if we study about these drugs through the book, so these drugs are very closely related as the mustard agent. That's why they can call mustard gas or nitrogen mustard. And they are cytotoxic because they are alkylating species. They, first of all, they undergo the hydroxylation process by this enzyme, and then they perform their action, very good for the non-Hodgkin lymphomas, carcinoma and breast cancer. Their mechanism is that they are used as alkylating agent. Both of these drugs, first of all, become activated, and they undergo the metabolism. They become activated. First, their activated form is phosphoramide mustard, and second is acrolein. Now this phosphoramide mustard, this react with the DNA and do the cytotoxicity, break our DNA. So how, next we move to the next page. Okay, next they have given the pharmacokinetics of cyclophosphamide. It is given orally or IV, while isophosphamide it is given through IV route, and their adverse effects are like hemorrhagic, cystics, nephrotoxicity, especially by ifosfamide is can be an hemorrhagic cystis. This can be prevented by hydration and by misna. Misna is cycloprotective thing. Myelosuppression and alopecia is their adverse effects. Okay, so this was the all things about the first group. Now we see this table in which they have given you the first drug, which is cyclophosphamide or isophosphamide, given oral or through IV route. Adverse effect are same. Their monitoring and their note is given that for good hydration can prevent the misna and these things. Use misna and hydration to prevent the bladder toxicity. Both are, these things are mentioned in both of these drugs. Now moving on to the second type of anti alkylating agent, which are the nitrosoureas. In nitrosoureas we have two drugs again, carmistine and lomistine. These two are closely related with it, that are primarily mean for the brain tumor. So now you just have to remember this, that all the nitrosoureas, lomustine and carmustine, they are very important for the brain tumors because they can cross blood brain barrier. Okay, that's why we can give them into the brain cancers, and they can easily travel at that site and perform their action by alkylating the your DNA. Mechanism is almost same. So their adverse effect is that they can cause myelosuppression, pulmonary toxicity, and hepatotoxicity. So these things can, and low mastine should be administered on empty stomach. So this is the specific thing for low mastine. Now see the table here. Carmistine and low mastine, they can be called as B, C, and U, and low mastine can be called as C, C, and U. IV and oral route is given. Adverse effect are the same, and the special note for them is uh, that your car machine, it is also available in the implantable wafer, and the low machine have to administer on the empty stomach. Okay, on the next page you have given the pharmacokinetics of it, which is just similar as I have told to you earlier, that one of them is given orally, another one is given IV route, and adverse effect we have discussed all of these things. Just things to remember about the cyclophosphoramide and isophosphoramide is that they are the mustard thing, and they will activate, and the phosphoramide mustard will react with the DNA and alkylate them. And next thing, the important thing that you have to remember about the nitrosoureas is that lomistine and carmistine both are very good for the brain tumors because they can travel through the blood brain barrier. Next thing, next important type of alkylating agent is decarbazine and temolامide. ठीक है, these both are the one of them is given through IV route, other is given through the oral route. Their mechanism is that they require the activation, they activate into active metabolite and temozolomide. It doesn't need any CPY 450, and it cross the blood brain barrier also. These drugs can be used in the treatment of brain tumors, melanoma, and metastatic melanoma. Adverse effects are just same. Myelosuppression is the adverse effect that is occurred through every drug, while nausea, vomiting, and these kind of adverse effects are done through every drug. Okay. Now they have written to you. First, they will transform into the active metabolite, which is methyl terrazino, such a long name, MTIC, simply its metabolites it. So its active metabolite name is MTIC. The metabolite is responsible for the activity of forming the methyl carbonyl ion with the DNA molecule. So it react and form this ion, and while your dark carbazine, it has ability to methylate the DNA. It add the methyl group into the DNA on the sixth position within the guanine uh, pyrimidine, used for treating these kind of lymphomas, and all the things are given here. Temozoline can go to the blood brain barrier and perform the actions there. Some other alkylating agents are also given to you in this paragraph, so I am telling the shortcut of them. The first agent that you can give is the melphalan. Melphalan is taken on the empty stomach. IV and oral route is it is for use for the multiple myeloma, and monitor the blood count due to variable absorption. So this is the melphalan. Next drug that can be used as a alkylating agent is the chlorambucil, which is taken with the food, orally taken. It is good for the chronic lymphatic leukemia, and it can cause hematology toxicity. Next drug is busulfan. It is alkyl sulfonates. IV route is given. It is used for the chronic granulocytic leukemia. And one of the major adverse effect of busulfan, which is very very important, is that it causes pulmonary fibrosis, which is called as bisulfan lungs. It causes lung fibrosis, make the lungs thick and hard. So these were the important things about the alkylating agent. First we have studied cyclophosphoramide and isophosphoramide. Both are the nitrogen mustard gases. They activate into the activated form through the liver enzymes, forming isophosphoramide mustard, which react with the DNA alkylate. Next drug included in it is the nitrosoureas, which is lomestine and carmestine. Both are very important, and they will react with your DNA. They are good for the brain tumors, so they can cross the blood brain barriers and perform the actions there. The next drug are the dacarbazine and tilmozolinide. Both are very good. First they activate into mtic, then they act, while tilmozoline don't need activation and it can go to blood brain barrier. That's why tilmozoline can treat the brain tumors, while they are good for myelomas. Melphenol, which is another agent of alkylating agent, it is used for multiple myeloma, not very important. Next is the chlorambucinol, which is used for lymphatic leukemia and such things. While busulfan is important because it is alkylating agent that is used for chronic myelogenous anemia, and its adverse effect is important because they can cause a pulmonary fibrosis. So these all they have written here. Like other alkylating agent, these agents are called as leukemogenic because all these three agents are treating the cancer of lymphatic system. That's why we can call this leukemogenic. So melphenolate, chloramphenol, chlorambucil, and busulfan. Three of these drugs are treating the cancer of leukemia. Dacarbazine is treating the cancer of melanoma, and telmazoline is treating the brain tumors also. Nitrosourea is treating the brain tumors, and while the cyclophosphamide and isophosphamide both are treating breast cancer, sarcomas, and non-Hodgkin lymphomas. So this was all about the alkylating agent. I hope that you will understand whole lecture. And if you find this video useful, then please subscribe to my channel, like my videos, and also press the bell icon so that you can get the notification of all the videos that I will be uploaded. Thank you so much. Meet you soon in the next lecture. Allah Hafiz.